Okay, okay, yep, says I'm live. Alright, hello everyone. I'm back. Hopefully, wait, just checking if it's picking my audio. Hello? Yep, alright. I'm gonna, my dad is like right next door, so I don't want to be too loud. I just hope you can hear me right here. Right, so before I start, I'm doing more biology and hopefully literature work as well. I'm doing more of that. But before I talk about that, I want to quickly talk about my previous stream. So I left a long description, pretty much explaining it. But just to re-explain, I had bad internet in the previous live stream, and what happened was I was using my hotspot as an internet, but what I forgot was if you like have more devices connected to the same hotspot, the hotspot starts getting really bad. So as a result, lots of the stream was cut out. I think like 26 minutes was cut out. I don't know, I forgot. But yeah, what I'm really annoyed at is at near the end. That's when like interesting thing happened. Again, it's in the description of that video. Anyways, let's go straight into it. So why am I streaming today? Well, I have biology homework today. No, I have biology test tomorrow. I have a biology test tomorrow, and I have not studied at all. So yeah, now's never then. Yeah, it's 9 o'clock. I was supposed to start like 2 hours ago. Didn't start. Many things happened, but I didn't start. But hey, you know what? Who needs to sleep anyway, right? Am I right, guys? Who needs to sleep? Again, I need to quickly check because I've discovered that if this says like poor, like poor is good, but like if it says like something along the lines of like no data, that means it's not recording the thing. Sorry, I need to quickly blow my nose. Alright, let's get into this. Let's get straight into it. So, what was I going to say? Yeah, so my last biology test went fine, actually. I'm surprised that I could actually answer the questions, which was amazing. So, clearly this has some worth to it. Streaming this study has some worth to it, even though no one's watching. which is good. Okay, so let's get straight into it, Let's get straight to it. So we're going to talk about biotechnology. Now, biotechnology is... I have no idea. I have no idea what biotechnology is. Oh, I saw a new document just to start fresh, So I don't know what biotechnology is. So I'm gonna search that up because that's how you find the meanings of things, right? You Google them. How else are you gonna find out meaning? So Google definition is exploitation of biological process for industry and other purposes, especially the genetic mutation of microorganisms. I have no idea what that is. No. Okay. Well, guys, turned out Googling was not a good idea. Anyways, so the plan of today is I'm gonna follow out through this PowerPoint. There's only about 21 slides, so it's not as bad as other slides that I've had, and I've got videos to watch, and my teacher has also set up an education perfect for me. Teacher's really nice, like really good teacher, by the way. Like the thing about this teacher is you can't. Oh, done it. Oops. The thing about this teacher, the teacher I have, is you cannot, like, if you fail his classes, it's your fault because it's because you didn't put the work into it and not just thing. Oh, where did my thing go? Okay, no, sorry guys. Just got. I don't know why that happened. Oh, this is. I'm so good. I'm so good at streaming, guys. Look at this quality stream. Alright, sorry. I'm back. Okay, live streaming. Stream health's good. That's good. Alright, let's get straight into it. So we're gonna go through this, watch the videos, go to Education Perfect, and hopefully do literature homework at the end. So yeah, literary homework. Let me just quickly read the novel The Crucible. If you've read it, that's cool, but most of you probably haven't read it. All right, so let's get straight into it. Let's get straight into it. Anyways, let's go. So, biotechnology. Using biological cellular processes to make products useful to humans. It also includes genetic testing, gene manipulation, cell replacement therapy, gene therapy, education. These processes all require different biotechnology techniques. It is these techniques we will focus on. Cool. Okay, so there are techniques. These are the processes. And I think, yeah, we need to focus on just biotechnology, I hope. Wait, where is my... when he's referring to page 108, he's referring to our science book. So let me just quickly grab that out. So let me just quickly skim through it. Let's see if I can read it and then explain it to you. If not, then that's a problem. That is a problem. Oh my gosh, that's so many words. Wait, genetic therapy doesn't have much. Gene therapy aims to treat or cure genetic abnormalities by replacing faulty genes with healthy ones. Yeah, I get that. And cell replacement is stem cells are individual any source of loss of cell replacement can be generated in most widespread. Stem cells are okay. I think it's just just replace it. Basically, it's about like to summarize it. It's like if there's a faulty gene, then you can replace that gene with another good gene. And if you have a faulty cell of DNA, you can replace it with a stem cell, which is something like people make in the labs, and they replace with. Oh, DNA. All right, I need to quickly revise this. Okay, so DNA, right? DNA has this. Should I just? So this is the. Where is it? Okay, there's a sugar phosphate. Yeah, here we go. This is the sugar. So there's a sugar molecule and a phosphate molecule, but I think just to focus on has A, C, T, G. So A is isotin, adenine. Wasn't even close. Adenine. C is cytosine. I really should know this already, but hey, I'm stupid. Thymine. Oops. Oh, sorry. I thought my keyboard froze again. Last time it froze, my keyboard froze and couldn't type, but that luckily was at the end of the stream. It was just lagging. So that's thymine, and this is guanine. G is guanine, and then I believe when it comes to RNA, it's exactly the same, but the T for RNA, it's URACLE. Now, in terms of pairing, blue always goes with green. GC and AT. It's always GC and AT. It's always GC and AT. Pairings, it's GC and AT. Okay, go. Okay, so that's a brief summary of all the DNA. Let's get into like the actual biotechniques. Okay, biotechniques. Need to know what is it, how is it done, and what can it be used for. All right, here we go. What is it? How is it done? How is it done? And what it can be used for. What can it be used for? So we will be answering these three questions for each technique. We will be for these four things. So DNA sequencing, gel electrophoresis, polychain reaction, and recombinant DNA technology known as genetic engineering. So we will be needing to answer these three questions around 4 times. I think what we can do for each one is, okay, so let's go to the first one. All right, here we go. First one is bold, underline. Let's make this bigger. Polymerase chain reaction, or just PCR for short. Cool. Hopefully it's poor. It says it's poor, but hopefully it's still recording. As long as it's not, what was I gonna say? As long as it's not deleting my stuff, it should be fine. Okay, when when it says offline or no data, that's when I should be serious. Serious issue that needs attention. I don't know how to fix this. Oh my gosh, Siri could hear me from here. Just wait, I need it. Just get. What are you talking about, Siri? Sorry, it's just my phone. Sorry, just distracted. All right, let's go. A cycle that is repeated in order to generate multiple copies of a DNA fragment. So that would be genetic testing. So, genetic pollen amplifying a DNA sample. Okay. All right. So what is it? How is it done? What it can be used for? It can be used for multiplying and amplifying. Does it say amplifying? Okay. It can be used for amplifying a DNA sample. Multiplying on an DNA sample. Did it say sample? I'm not sure. Oh, I keep having to go back. Amplifying a DNA sample and generating multiple DNA fragments. Multiple copies of a DNA fragment. Okay, what do we need for it? Okay, I just realized I probably need to underline these things so that it can. What is it? But what is it though? What is it? What is it though? A cycle that is repeated in order to generate multiple copies. A cycle. A cycle? A cycle. All I know is it's a cycle. It's a cycle. How is it done? All right. Alright, so first to discuss this, we're first going to need, what do you need to do to cause a polymerase chain reaction? Well, first you need ingredients, right? You need ingredients, of course. So what do you need for ingredients? I do have other notes besides this that I wrote down during class, so I was kind of paying attention. But guys, please give me this one. Wait, did I? I'm not sure what I wrote it in my book or somewhere. Let me just start. So my room is a big mess. You can probably hear just from the audio, but it's a big mess. Okay, how are we doing for time? Been 11 minutes and we just polymerase chain reaction. We are doing so well. Am I right, guys? Okay, this doesn't want to connect to the internet. While I'm waiting for this, I just want to talk about tomorrow. I might there's this. I think I mentioned this before, but there's this short story competition that I want to compete in. It's the entries are due 14th of August, and now it's the 10th of August, and I haven't even written my story, which is not good. But I want to at least write it and submit it. It doesn't even have to be good, but I just want to try, and it's worth the stream. So I want to stream a whole thing until I finish the story, and and just like that. Anyways, here we go. Now, what can we do about this? Okay, so it's 9 o'clock. I should be able to sleep like 12. I don't know. I think I have to wake up early tomorrow though. But you know what? Who needs sleep? Seriously, who needs to sleep these days? Am I right, guys? Am I right? Anyways, let's just DNA pause me. So, ingredients cause DNA polymerase, which kind of makes sense because that's the name of the thing with DNA primers. I don't know what those are. DNA template to be replicated, obviously. DNA template to be replicated, obviously, because if they're copying something, they need the original first. And the DNA nucleotide, and they have in italics, okay, temperature change. So at one point they need to change the temperature, not an ingredient, but compulsory. All right, cool. So those ingredients, I had notes, but my oh no, sorry, my computer is not getting the internet. I had other notes. I hope I can't find them. I'm so organized. Can you tell, guys, how organized I am? Okay, wait. Let me get my science book. Science textbook. Sorry. Where is it? Oh, here it is. Okay, so what did I write down for this? Biotechnology. Oh no, I think I put it on my Chromebook. That's not good. Everything is on my Chromebook. And my Chromebook is very slow. It's working very slowly. What are we gonna do? Should I watch the... Okay, I'm gonna go through... How many... Yeah, okay, so there's 1, 2, 3, 4. There's like... Wait, no. 1, 2, 3. There's just 3 more... Just 3 more things I have to do. And, oh my gosh, the internet is just not working. Which is weird. That shouldn't be the case. Could I possibly go into... No, darn it, I can't do this. That's not good. That's not good. Sorry, I'll be right back. I need to quickly check if I just need quickly, I just quickly need to go and check something because my internet, I need to move closer to the internet for it to work. I'll be right back. Sorry, I'm back. Sorry, just need quickly. Okay. Okay. Cool. Cool. I'm coming. I'm here. I'm back. Okay. I got it to load finally, which is good. So I wrote here DNA sequencing. Okay. DNA profiling. DNA. So DNA sequencing means determining the order of nucleotides, poly chains, amplifying a DNA. Okay. PRC is the starting point. I wrote. Oh wait. I should probably get out of there. PRC is the starting point. The starting point. I don't know what that means. Starting point. A cycle that is repeated to order. Yeah, I already wrote that. Okay, ingredients. Okay. So I wrote here. This is what I wrote in my notes. Enzymes that does the replicating and copying. Okay. So this is what actually yeah, it's pretty straightforward. Don't need to explain that. From bacteria, which is, and that's you get Tay-Sachs polymers from thing. Directing the, it's like, and so this goes at the ends of this thing, of the thing. Directing the DNA polymerase, like the direction it goes in. That's kind of obvious. Nucleotides, so they can be added in strands. I wrote so they can be added and build new strands, whatever that means. So that's what I wrote down. Not sure if that's going to be helpful or anything, but that's good, I guess. Alright, let's keep going. Alright, the method. So, combine the aptitude. So, combine ingredients in an aptitude. Combine method. Method, question 1. Here we go. Combine. Is this working? Please tell me it's working. Alright, here we go. Combine. Sorry, it was lagging right there. Combine. Sorry, do I have anyone watching? No, no one's watching. That's good. That's good. That's always good. That's always good to see. It's always good to see. Alright. Combine. Off time. The ingredient in it. Combine the ingredients in an aptitude. Cool. And then denature. Oh yeah, denature. Denature. I know what denature is. Hard to explain, but heat the contents to 90 to separate the two germs. Screen. All right. Heat the content to 96 degrees to separate the two strands of DNA. Yeah, the two strands of DNA. So mix ingredients, heat them up to like really high so that they can be separated. I pretty much just repeated myself. Annealing. That's new. I don't know what annealing is. In fact, I actually should know because we went through it in class, which is not good. I keep going to the next one. Cool the contents to allow the primer to attach to DNA. Alright, cool the template. Cool the content. Sorry, not the template, the content. Cool the content. Cool the content to allow the primer to attach to the DNA. Attach to the DNA template. Cool. That's form. Extension. Okay, so each of these steps have a name actually. So we have to heat it again, heat the content. So it's pretty much a bunch of heating and cooling basically. 72 degrees. 72 degrees to allow. I'm just copying and pasting here, but at least I'm typing it so it goes in somewhat in my memory. Hopefully it goes in memory. I highly do not recommend that you do this loosely. Last minute study last nights don't loosely work. But hey, some study is better than no study. Am I right, guys? Oh, well then. I do hope my dad isn't hearing me. My dad's next door, but he's on the phone, so I'm not sure. I hope he probably doesn't hear me, which is good, but I hope he doesn't. It would be very awkward if he was listening. To synthesize, and to synthesize means just to create, right? Yeah. 72 degrees to allow the DNA to synthesize new strands using the DNA nucleotides. To synthesize. So synthesize means just to create new DNA strands. So basically, it's very straightforward. New strands of DNA to new strands of DNA and using the nucleotides. Using the nucleotides. Repeat cycle. Okay, that's pretty straightforward. That's actually pretty easy to understand, actually. I'm not sure why. It always sounds so complicated when I hear in class, but when I go through it myself, very easy. I just don't know what it looks like. That's probably a big problem. I don't know what it looks like. I don't know what this is. What is this? Oh yeah, so in my previous stream I talked about like why like DNA goes in different directions. 3 to 5 direction is 5 to 3. So 3 is always going to 5, and 5 to 3. Okay, this is about direction. Number representing carbon. There are 5 carbons in the thing. Yeah. So the direction of the DNA. So like each in the DNA, there's like 5 sugars. Yeah, in the sugar there's 5 carbons. Sorry, and each carbon has a number, and like 1, 2, 3, 4, 5, 5. And depending on how they're positioning, at like in one side there's number 3 in one side is number 5, so, and then I believe it's the direction's always 3 to 5 and 5 to 3. I'm not sure which one is it. I'm pretty sure it's one or the other. I'm not sure. Okay, so here's PRC process. We have the denaturing. Okay, so here's a sample. We separate it. using denaturation, which you have to get to like 96 degrees. Then we have to cool it back down to 55 degrees, which is primer annealing. And then we have primer extension. So basically just annealing extension. I don't think we need to learn about primer. Back to 72. So to 90, to 50, then to 70. 96, 55, 72. So primer attaches these. Now these things, I believe, are the ones that this directs the DNA strand. So the annealing to cool down attaches these primers to the DNA strands. Extension 72 degrees. This is when the replication begins. Yeah, see, so this thing, the replication is going that way. Here it's going that way. So it's two different directions. Result after one cycle of DNA molecules doubled. See, cool. And repeat. I'm gonna take a picture of this and put it on my thing, on my document, so that I can have a reference. Cool. How do I insert? Yeah, insert. Here we go. Image. Load from computer. Desktop. All right, cool. That's pretty straightforward. All right, one out of 4 done. That was quicker than I thought. That's bigger than I thought it was gonna be. That's very big. Oh, well then. Okay, how's it done? What can be done? Okay, what can be for multiplying, generating for amplifying. This is amplifying. Amplifying a DNA sample. Generating, amplifying a DNA sample. Alright, let's go. Let's keep going. I think that's it. Chain reaction. Yeah, so it goes into 1 to 2 to 4. Wait, no. Can I count? Yes, I can count. 4 to 8. Wait, no. 4, 2, 4, 6, 8. Yes, I can maths, guys. It's okay, guys. I'm good at maths. Trust me. Yeah, so it just keeps doubling. So if we were to repeat the cycle again, we can like eventually have lots of it. If each strand is then doubled, or so on and so on. I think it moves on straight to the next one. All right, so now I want to, which one? Put this one. All right, so how long is this one? I might want to, it's only 4 minutes. 4 minutes. That's good. We can deal with that. We can deal with that. We can do that. Okay, again, sorry, I forgot. I keep saying that I need to do the audio. Like, I haven't done the audio, so you're just hearing it from my
In this video, we're going to talk about something known as PCR.
Shut up. Sorry.
Stands for
Alright, I'm gonna skim through this. And hopefully, how much? I have 62%. Oh my gosh, that's not enough at all. I'll put this here. No, actually, I'll put this here. There we go. And now, let's watch the video. You may want to turn down the volume just in case it hurts your ears.
Nice. Chain reaction. So let's look at the process of PCR. And let's start by looking at the history of PCR. PCR basically starts off with some DNA that you're trying to amplify. You're trying to make more copies of the DNA. And you start off.
Sorry, so amplifying and copying is basically the same thing.
A piece of DNA, it's got 2 strands, and in order to denature the strands, you apply heat. Now, the problem is that a lot of enzymes that would normally, once you have the 2 strands separated, a lot of the enzymes that would normally then copy the strands and make double-stranded DNA would denature.
Wait, what? Wait, that would normally
once you have the 2 strands.
Once you have the 2 strands separated, a lot of enzymes that would normally then copy the strands and make the double-stranded
DNA. And make double-stranded DNA would denature. They wouldn't work anymore because
they wouldn't. Oh, okay. Because you want the enzymes that are copying the DNA, you still want those enzymes to work so they can copy. Unfortunately, if they denature, they can't work. That's also another reason why you cool them down again. I think that's why you cool them down. Oh man, I just scratched something off my screen and I don't know what I just scratched off.
Basically destroy them. So scientists were able to isolate a bacterial species that actually grows in the geysers in Yellowstone National Park, which is an extremely hot environment. So the bacteria was something known as Thermus aquaticus.
I don't think I need to know that.
The species of bacteria that basically had a DNA polymerase, and we call it TAC, or Thermus aquaticus polymerase. And this polymerase is able to withstand the high heat needed to denature the DNA strand.
Oh, okay. So the enzyme that replicates it is from a bacteria. It's from that bacteria, and the reason why it can withstand the heat Okay, wait, never mind. So forget what I just said before. The reason why that's not why they cool it down. The reason why they heat it, or the reason why they use this type of enzyme, is because normal enzymes would literally denature with the DNA once you heat it to like 96 degrees. But if this bacteria's enzymes specifically are made to withstand heat, so I see.
So if you apply Taq polymerase to the now denatured single-stranded DNA, it's able to make a complementary copy of the DNA. Able to copy it. So basically, this is the overview of PCR. Now let's look at the specifics. So you start off with some DNA, and you apply heat and denature the DNA. So now you've got two strands. What you next need to do is you need to add a primer. So you need to add a little primer, and the primer gives the Taq polymerase a starting point. So the Taq polymerase comes in and basically is able to make the complementary strands.
Okay, so they elongate. What does elongate mean? Okay. So that's what primers do. Guys, you got what primers do, right? Do you understand what primers do? All right, please explain to me what primers do. So again, so that's the starting point, obviously. So, as you said, that's where the copying of the DNA starts, and where does it go, left or right? That depends on the primer. I get it.
In order for the Taq polymerase to make the complementary strand, you also need to add in nucleotides. So you need to add nucleotides, and you also needed a primer. So basically, heat acts like helicase, which normally in human DNA would split the double-stranded DNA up into two strands. And the Taq polymerase basically acts like our own DNA polymerase that normally would replicate the DNA. So basically, if you repeat these steps, so you heat, you denature the DNA, then you reanneal the DNA, allow this elongation process to occur. And if you just keep repeating that, eventually you'll have a whole bunch of copies. So here you start off with one copy of double-stranded DNA, here you have two copies. If you repeat it, you're going to have 4 copies, and so on and so forth. So you basically get this exponential increase in the copy number of the DNA. And this is really useful in order to create massive amounts of a particular DNA sequence. So what I want to now talk about is I want to look at the exponential growth that is a hallmark of the PCR reaction. So after we do PCR one time, so we start out with double-stranded DNA, we add heat, it gets denatured, and now we have 2 strands of double-stranded DNA. So this is one round of PCR. If we do another round, we're basically going to have each one of these denatures, and each one after we add Taq polymerase and after we add the primer, we're going to have 4 strands. So this is after round 2, we have 4 strands. And after round 3, we're going to have 8. So we go from 1, 2, 4, 8. After round 4, we're going to have 16. After 5, we're going to have 32, and so on and so forth. And basically, after just a few rounds of PCR, we're able to have thousands and thousands of strands of double-stranded DNA. And that's the power of PCR, is that you get exponential growth. So you start out here, and then you really just rapidly get an exponential amount of growth and an exponentially larger amount of DNA that you're interested in.
Cool. Pretty much revision from what we just learned. But that's good to... Repetition is the best way to learn things, am I right, guys? Especially when I'm not writing this down. When I write down things, I'm aware that writing it down, like, you learn it better when you write it down. But, so that's why I gotta work, like, put more thought into it, just so I can learn better. So twice is the charm, I guess. I have no idea what I just said. Oh well. Let's move on to verses. It's just when I don't know, I can't write it down, because if I write it down I procrastinate. And if I procrastinate, that's not good, is it now? So what I'm saying is typing is better than just doing no work. Did I spell this word correctly? Extrophoresis. All right, cool. There's a picture. Oh, pictures of my weakness. No. Okay. Oh wait, no. Let's look at the overview. Wait, how many? There's 1, 2, 3, 4. Okay, so just 3. Okay, so just 3. There's just 3. There are just 3 slides with this, which is good.
Tomorrow has to be early though, maybe.
Yeah. Okay.
You know that.
Yeah.
6:30 wake up, yeah.
Okay. Yeah, sure.
7:30 come back. 7:55
Yep.
has to be in the school.
Okay. Yeah. Sure. Yeah. Sorry, I have to wake up early tomorrow. This is exactly like last time. In my last stream, I also had something due on Tuesday, and I had to wake up early the next day, which was not a good feeling. I did not work though. I did not feel good the next day. You know, my very first stream was me. Is it good? Is this good? All right. My very first stream was me. We're already up to half an hour. Oh my gosh! What the? Oh my gosh! Wow. Okay. Anyways, my very first stream was me doing an all night on a assessment that I was supposed to work harder on. I did better than I thought it was, but yeah. Anyways, I need to look at this overview. So it's just three. Alright. Let's just do this. Used to separate DNA fragments according to their size. DNA samples are loaded into the wells. So basically, what is it used for? Separating DNA molecules. Separating DNA fragments. Separating DNA fragments. Man, I should have started earlier, man. I should have started like 7, but I don't know, man. Kept procrastinating. I shouldn't have started 7. This was not a good idea. Oh well. Let's do it. Let's do this. Let's just keep going. It's separating DNA using size. Cool. How? Just on curiosity, how long is the jail? 7 minutes. Oh, that's 7 minutes. No, that's 8 minutes. That's pretty much 8 minutes. How long is this one? 3 minutes. Okay, so Sanger method is short. Gel electrolysis is basically the same thing. I need to wait. Can you see me when I type in my password? Yeah, you can. Right, cool. Hopefully that doesn't. Okay, used to separate DNA according to size, DNA samples are loaded into wells, indentation at one point, end of the cell, and a current electricity is applied through them through the jail. Okay. DNA fragments are negatively charged, so they move towards the positive electrodes. Because all DNA fragments have the same amount of charge per mass, small fragments move to the gel faster than large ones. When a gel is stained with a DNA binding dye, DNA can be seen as bands, each representing a group of the same size DNA molecules. Alright, cool. So this is the gel, that's the wells, and then that's where, so this is where the DNA fragments are going, and they would go this way towards the positive, and the smallest ones are faster. Okay, I get it. Cool. Uses. Oh wait, this is what it's used for. Okay, never mind. Okay, so what is it? What is it? How do I answer this question? What is it? I know how it is done. I know what it's used for, but how do I say what is it? Okay, what's that amount to? I'm up to 21. Alright, cool. So let's just go. How is it done? I'm just saying. Used to separate DNA. Okay, probably there's more on the thing. I know I'm repeating myself here, but at least I'm. Can you hear me scratching? I'm scratching. I hope you don't hear that, but I'm scratching. Hope you don't hear me scratching. That would be weird. Okay, so let's just, I don't know. What's this? Okay, so that's for uses and that's for overview. So this is basically the method, really. This is basically the method. So let me just put that down here. Let me just start here. All right, cool. Used to separate DNA. Yes. Separates DNA fragments. What? According to size. Sorry, according to size. Question 2, why is it not doing the thing? That really annoys me. Wait, why is it not doing the thing? Oh, there we go. Alright, cool. Okay, so DNA samples are loaded into wells, indentations. So the wells, by the way, they're not actual wells. They like basically indent the gel. They make like a small hole in the gel, and that they call it a well, basically. So DNA samples are loaded into the well indentation at one end of the gel. At one end of the gel. Gel. Sorry, I could have sworn I heard something. At one end of the gel. Oh, wow, I had 2 views at one point. Oh, who knows? Okay, so DNA strands are loaded into DNA indentation at one end of the card. Sorry, I just could have sworn I heard something. Oh. It's like breathing, anyways. And an electric current is applied to pull. Oh, at one end of the gel. Sorry. And an electric current is applied to pull them through the gel. Pull them through the gel. Okay, I can picture it. I'm picturing it now. So basically, that's why they put an indent. So they don't want to go through the air, they want it to go through the gel. DNA fragments are negatively charged and hence move towards the positively charged electrodes. Because all DNA have the same amount of charge per mass, small fragment. Okay, all DNA fragments has the same, what's this? Charge per mass. Have the same amount of charge per mass. That means like if one DNA has this much charge for 2 grams, then every 2 gram DNA will have that exact amount of charge. But a 4 gram DNA would probably have less. No, would have less charge or more charge, or double or half the charge, basically. And therefore, small fragments move to the gel faster. Small fragments move faster than bigger ones, and that's how they separate them via size. Sorry, when a gel is stained with a, that's lagging again. Stained with a DNA binding dye. So the DNA fragments can be seen as bands. The DNA fragments can be seen as bands, each representing a group of the same size. Each representing a group of same-sized DNA fragments. Alright, cool. So so now that I've actually read through these, it's so helpful, these PowerPoints. I cannot express how helpful these are. If my teacher didn't make these PowerPoints, I would be dead. Anyways, gel strain with binding dye. How's it done? That's good. It said I had 2 concurrent views. I wonder who that was. Oh my gosh, what's up? Oh, Crispy Tofu, I know you. Oh, hello. Hey, hello. Hello, sorry. This is, if you don't know what this is what do you call it? A friend of my little sister who also has a channel. Go check him out if you want. Is he still watching or did he leave? It says, I think he left. Did he left? I'm not sure. I think he left. I think my boring studying made him go away quickly. I mean, I don't blame him. This is literally me studying. What entertaining is there? I wrote boring stream on my thing, right? Oh yeah, I think he left. I wrote boring stream. Yeah, I wrote boring stream. So people know what they're getting into when they click on this video. I don't know why anyone would click on this video, but hey, that's so weird saying that. Oh, he's back. That's so weird. I don't know whether he's back or not. Oh well. Let's keep going. Let's not get distracted now. So basically, to explain this picture, right, those are the indentation. This is the gel. You can see it from here. So the samples would be put inside these wells. Then they go towards this way, right? But because DNA samples are loaded into the wells, power is turned on and DNA fragments migrate through the gel, the largest and the smallest. So this is pretty much it. So I like this. I pretty much understand this. Is he still there? I don't know what he's doing. So I'm going to take a photo of this. I'm going to put it on my thing. I'm going to need to charge my MacBook soon, but I'll do that later. And what can it be used for? So we're almost done with this. We just need to watch that 7-minute video. Oh, we almost reached an hour. I just realized. No, wait, no. Still 46 minutes. That's good. Insert image. Upload for... Man, if I could stream every single day, I would be so smart. I'm not even joking. The thing is, my mom also uses this MacBook for teaching, so it's quite complicated. Choose. Cool. Here we go. All right. Can't even read that, but oh well. I pretty much get the gist of it. What's important is that I understand. The next slide. Pretty much. Sorry, I need to stop checking on this. The uses. We need to put dot points. What is it used for? Separating DNA to create DNA profiles. Right? DNA profiles, separating proteins by size and charge. So mostly separating protein. It can separate protein by size and charge. It can separate RNA fragments. So it's basically separation. Whenever I hear separation, I need to go gel electrophoresis. Separating, what was it? RNA fragments. Can identify gene mutations as what? Can it? Really? Can identify. So it can also identify. I guess, I don't know. Gene mutations such as cytic function, and Huntington's. It's very complicated. So I need to stop checking. Okay, that's the last time I'm checking. So I'm just checking if he's reading. Just checking if he's there. Used for DNA sequencing. Now I know what DNA sequencing is. Wait, did I skip one? Species. Okay. It can be used to determine the relationship between different species, and then used for DNA sequencing. So DNA sequencing, I believe, just means, what is DNA sequencing? Oh, sorry, dropped my glasses. I was scratching my ear and I dropped my glasses. DNA. Oh, did I write down DNA sequencing? I did not write down what DNA sequencing was. Okay, DNA sequencing. I said it before, didn't I? Wait. Man, I'm so dumb. What time is it? It's not even 10, which is good. We're halfway, which is great. I'm surprised I'm actually doing this much work. DNA sequencing is basically... What is it? Yeah, DNA sequencing at the end. They can be added. A cycle that's represented. DNA sequencing, genetic DNA sequencing. Oh yeah, here we go. Here we go. Sorry. Where is it? where am I? Okay, DNA sequencing, which is just determining the order of nucleotides. The order of nucleotides. Alright, cool. Alright, I think that's it. And that's next one. So before we go in there, again, we need to look through this. I'll look at this. This by the Amoeba sisters. Again, pretty much. I'm going to skim through this again because I already kind of doing it, but the revision is always good. So let me just. Oh, it's an ad. No.
No Wi-Fi?
No, not an ad. Yeah, I'm muting it. There we go. Alright, now I'm gonna just turn off. Captions are on turn off by did they write that, or is that just a YouTube thing? I don't know. Hopefully they didn't write that because we're not that stupid. No. Okay, sorry, I thought I deleted the thing. Command C. Go check out this channel if you want to, but there is that link if I ever want to revise. So again, I want to just go through this quickly. I'll slow down if there's something I look through and I was like, wait, what? Let's go. Oh, they did write it. They did write that. Captions are on. Turn off by clicking C at the bottom right. Updates on Twitter. Yeah, go follow them on Twitter, guys, if you're watching. Here we go.
When I was a young kid, I was really interested in genetics.
I wasn't.
I didn't really understand genetics. I kind of thought that when two organisms had a baby, the baby was just this blend of the two. Yeah, that's a misconception. But I really saw genetics in action with my guppies. Guppies are very easy freshwater fish to keep in an aquarium. But they have two things that I think are especially cool. They have live birth, which
means there are no eggs. There are no eggs.
There are no eggs, like many fish. And they have a lot of babies. They also eat their birth, so are very easy freshwater fish to keep in an aquarium. But they have two things that I think are especially cool. They have live birth, which means there are no eggs, like many other fish. And second, they have a lot of babies. They also eat their babies, but I don't think that's especially cool. So, you can see that.
I'm so dead. I'm so tired. I laughed at a joke that wasn't even funny. No offense to this channel. This channel's been really helpful. No eggs, live on. Variety of colors, lots of babies. Alright, cool.
Of my cool fact list. Anyway, when my surviving baby guppies grew up, they would have all kinds of cool traits. These traits were carried by their DNA, their genetic material, which is found in their body cells. But sometimes I would forget which mother was the mother of the baby guppy. Because there were several mother fish in the tank, and I wanted to keep track of inheritance in my guppy notebook. So what would have been very cool to have at that time? Some biotechnology. Biotechnology is the merge of biology and tech, and it's constantly changing. It includes topics such as PCR, cloning, and genetic engineering. It's also an awesome field. We're going to talk about one of the biotechnologies that could have, well, potentially, helped me determine the genetic relationships of my guppies. If, as a young kid, I had access to it, although it's becoming way more common in classrooms now. And that biotechnology is gel electrophoresis. Gel electrophoresis can be used to separate molecules based on how big they are, their size, and it's especially useful with DNA. Let's look at DNA real quick. So here is a guppy cell, here's the nucleus in the guppy cell, here's the DNA in the nucleus of the guppy cell, and if you were to zoom into the DNA, here is a nucleotide, which is a building.
Nucleotide, I forgot. So I need to lower down that voice. It's a bit loud. It's a bit loud, so I'm going to lower it down.
Block of DNA. See those phosphates in the nucleotides? They're a bit negative. Well, they contribute a negative charge anyway to the DNA. So if we look at this whole DNA here, it gives that DNA a negative charge. That's a big deal because gel electrophoresis.
Okay, so that's why it's negative. I didn't know why it was negative. I just went with why.
Which, again, separates molecules based on size, relies on the fact that DNA molecules negative charge. Okay, here's a gel electrophoresis machine. The point of the machine is to be able to have an electrical charge running through a gel. So here's the gel, typically made of agarose. Agarose is a polysaccharide polymer, which if you remember from our biomolecule video, polysaccharides are carbohydrates. Yeah, usually agarose comes from seaweed. The agarose gel itself lets the DNA molecules travel within it. One end of the gel has these holes called wells. The wells are where the DNA is placed into. The area of the gel where the wells are is negatively charged, and the area of the gel here is positively charged. So guess where the DNA will travel towards? Well, since it's negatively charged, it's going to travel to the positive side. So typically when you're analyzing DNA in electrophoresis, you use these restriction enzymes to cut the DNA up into tiny pieces. Restriction enzymes have the ability to cut up DNA in very specific areas, often related to the specific
Sorry, I'll be right back. I need to get a jacket. And it's raining outside, which is cool. Oh, gosh. You know, usually I would be asleep by now. But you know, since when does school let their students sleep? Am I right, guys? Anyways, man, I really need to stop talking to myself. Alright, let's go.
When you're analyzing DNA in electrophoresis, you use these restriction enzymes to cut the DNA up into tiny pieces. Restriction enzymes have the ability to cut up DNA in very specific areas, often related to the specific DNA bases, making restriction enzymes very useful in biotechnology. So if I had baby guppy DNA and adult mother guppy DNA, and I want to compare them, then I would want to use the same types of restriction enzymes in both DNA samples. If I use the same type of restriction enzyme, it should be cutting the DNA at the same identification points in the DNA samples. However, unless the mother and baby guppy are clones, and they're not, those pieces that result after the restriction enzyme is done with them may be differently sized because the DNA of the baby and mother guppy had some differences in the sequence of their DNA base.
Repeat that.
It's very useful.
Restriction
Positive side. So typically when you're analyzing DNA in electrophoresis, you use these restriction enzymes to cut the DNA up into tiny pieces. Restriction enzymes have the ability to cut up DNA.
That's right.
You
So when typically when
use restriction enzymes to cut the DNA up into tiny pieces. Restriction enzymes have the ability to cut up DNA in very specific areas, often related to the specific DNA bases, making restriction enzymes very useful in biotechnology. So if I had baby guppy DNA and adult mother guppy DNA, and I want to compare them, then I would want to use the same types of restriction enzymes in both DNA samples. If I use the same type of restriction enzyme, it should be cutting the DNA at the same identification points in the DNA samples.
So what did that say in the corner? Would likely need to use PR to have enough copies of another video. Okay, so before gel electrophoresis, the only way to actually make comparisons like this, you need PCR to copy. Cool. DNA primers, DNA.
Adult mother guppy DNA, and I want to compare them, then I would want to use the same types of restriction enzymes in both DNA samples. If I use the same type of restriction enzyme, it should be cutting the DNA at the same identification points in the DNA samples. However, unless the mother and baby guppy are clones, and they're not, those pieces that result after the restriction enzyme is done with them may be differently sized because the DNA of the baby and mother guppy had some differences in the sequence of their DNA bases. So the DNA samples both are cut up into multiple pieces by the same type of restriction enzyme, and then those samples are loaded into the gel. Sample 1 and sample 2. If we turn on the machine and let the DNA run through
what? Maybe I should not be watching this at full speed, but I'm learning things that I did not learn in the PowerPoint.
And I want to compare them, then I would want to in biotechnology. So if I had baby guppy DNA and adult mother guppy DNA, and I want to compare them, then I would want to use the same types of restriction enzymes in both DNA samples. If I use the same type of restriction enzyme, it should be cutting the DNA at the same identification points in the DNA samples. However, unless the mother and baby guppy are clones, and they're not, those pieces that result after the restriction enzyme is done with them may be differently sized because the DNA of the baby and mother guppy had some differences in the sequence of their DNA bases. So the DNA samples both are cut up into multiple pieces by the same type of restriction enzyme, and then those samples are loaded into the gel. Sample 1 and sample 2. If we turn on the machine and let the DNA run through the gel, the DNA moves towards the positive side. But some pieces of that cut up DNA will move faster or slower than other pieces. Longer DNA pieces tend to have a higher molecular weight. They take more time to make it across the gel when you compare it to shorter DNA pieces, which move at a faster rate. So what you end up with is that these DNA fragments spread out, with the longer pieces closer to the wells and the shorter pieces closer to this opposite side of the gel. These are called DNA bands, but to see them, you usually need to stain the gel itself and view it under a UV light. Now let's compare the DNA bands in this hypothetical, simplified guppy situation. The bands aren't going to be identical because these fish are not clones, but I can compare how similar the bands are and compare that to other mother guppy samples to look for relationships. Let's say that we have 3 mother guppy samples to view, and these are the only possible mothers from the fish tank. Which one appears to be the most related to the offspring in this case, and has a high likelihood of being the mother? Well, this one. But we can't be 100% sure with this. It would be helpful for me to know.
Hypothetical, simplified results. No guppy DNA was electrified for the video.
Father guppy sample 2, because this will give you more insight. But if these are the only fish in the tank, it's a very high likelihood with this case.
Until they ate him. Fun fact, I can eat it.
Also, you can use something called a DNA ladder. You can buy them from various science material distributors, but a DNA ladder is not what it sounds.
Changing light bulbs, helpful identify.
It's basically a sample that has known fragment sizes. So if you run it in the electrophoresis machine, you already know the fragment lengths. Let's say this DNA ladder only had 3 bands, which is not usually the case. Since it's a DNA ladder, the base pair lengths are known. They are 500 base pairs, 1,000 base pairs, and 1,500 base pairs. Think for a minute. Where would they fit in?
Okay, wait, sorry. Okay, I'm gonna guess here. 500 near the negative, and 1,500 near the positive, and 1,000 in the middle.
It would look like
No, I was wrong. Hey, I got 1,000 right. Oh my god, I'm so stupid. Okay, 500 is for the short term.
you can use this now as a reference to give estimates of how large the other fragments are when they're run alongside it. And if you
Oh
want
yeah,
to
true. The bigger it is, the longer it would take. Therefore, it's shorter the negative side. The shorter it is and the smaller it is, it would go to the positive side. Therefore, there would be less base pairs in the shorter one, more base pairs, I think, because the DNA is larger, hence they would count as more DNA. At least that's what I think.
estimates of how large the other fragments are when they're run alongside it. And if you want to be closer to the value, you can use a semi-log graph, something to look up. So why do we care about gel electrophoresis? It's not likely I'll be actually using this.
Can we just get a nice toast instead? Oh, wow. I was gonna make a joke to reply to that joke, but I just realized I'm not a funny person, so I'm gonna keep going.
My guppies anyway, right? Well, perhaps. But gel electrophoresis is often a step used in determining relatedness with different species, which helps scientists better classify organisms. It's also used as part of DNA fingerprinting. DNA fingerprinting is a way that one can identify someone's DNA, which can be very helpful if you're trying to solve a mystery involving a crime scene. If you have a DNA sample from a crime scene, you can go through the steps of gel electrophoresis to compare it to the suspect DNA to see the likelihood of a match. In fact, you can take the results from gel electrophoresis and isolate genes of interest by something called southern blotting. Definitely something to look up if you're curious. Gel electrophoresis is one of many awesome tools in biotechnology. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
No, I do not. I prefer not to be curious, but they say curiosity kills. I really need to stop making jokes, man. I for some reason I always have this urge to make jokes and make people laugh, but I always fail. I always fail at it. Anyways, okay, sorry. I need to. What was I gonna do? I was gonna do something, but I forgot what I was gonna do. So I'm gonna sit back down. Okay. What was I gonna do? I forgot what I was gonna do. Should I just move on, or should I? Okay, the things I didn't pick up from this. Okay, I actually remember this. You kind of remember. You probably it already explained it to you, but I actually remember. I actually now remember learning this. All I need to know from this video that I learned new was there are enzymes. You need to use the same enzymes to cut it. You need the PCR to multiply it first before using gel releases. Then you need like what was the other thing that we did? DNA ladders, and then 1,500 base pairs is closer to the negative, 500 is closer to the positive. Alrighty then, let's keep going, shall we? Segment it. Now, I'm pretty sure the rest of the slides literally dedicated to just DNA sequencing. Oh my gosh. Okay, so I was wrong. There's not 4, there's just 3. So, but it's gonna be long ones. So you might as well count as 2. Am I right, guys? I need to stop saying that. Okay. I just realized I need to go to the bathroom. So I'll be right back. And I'm on 26%. Probably need to charge that. Oh boy. Just need to quickly. Alright, I've got into 1 hour, which is cool. Which is great. Please wait a second. I'm gonna go in my bathroom break. I'm also gonna charge this, so excuse me for a section. Excuse me for a sec. Da-da-da-da-da. Sorry, I'll be back. I just need to go to the bathroom. And I'm also gonna charge this one. Sorry if the noise. Very noisy, but gotta. gotta go. It's actually charging this. Am I charging? Cool. I'm charging. Alright. See ya. I'll be right back. Okay, I'm back. I'm back, guys. Hello. Is this still working? Okay. Alright, cool. Now, DNA sequencing. Is that the final one? Just delete that. The Sanger method. DNA sequencing, pretty much. So let me just quickly look. Okay. Sorry, let me just quickly. There's like. Okay, so two of them are like pictures. The last two is okay. So the last two questions are just the last two slides are just questions, and two of them are just pictures. So therefore, there's technically only like 4 slides to go through. Yeah, cool. That's cool and everything. 1 hour and 40 minutes. Hi, sorry, I'm so tired. I don't know why, I just started getting very tired. But surprisingly, we're almost done with biology. We just need to do literature and we're done. That would be great, won't it? Alright, cool. Let's keep going, shall we? Alright, here we go. DNA sequencing. Sanger method. Sanger method. Should I watch the video first? Sanger method. Here we go. I'm so tired. I really shouldn't. I really need to stop doing this. I really need to stop doing this. I need to like actually like my second stream was cool because I was just casually studying. Nothing was due the next day. It was all great and everything. And then, I just had to mess it up. There's a video here, but I'm not sure whether that's the same video as this one. Is this also? This is only 3 minutes. So I might also, what is this? What is this? Khan Academy? It's an article. You're joking? Is this not a video? If it's not a video, I'm not interested. How's the sequence in nucleotide placed is determining. Okay, cool. That's cool. How much percent is my phone? Alright, cool. Okay, I want to do the same. Okay, this is not showing anything, so I'm gonna cancel that. Alright, cool. Let's go. What is it? What is it? Well, that's a great question. What is it? Okay, it's the process of determining the sequence of nucleotides. In brackets, they have S's, T's. So like the S means just lots of them, and then CS. Alright, cool. In the piece of DNA. In a piece of DNA. Cool. It also, what else is it? It's also it's lagging. In Sanger sequencing, the target DNA is copied many times, making fragments of different lengths. Fluorescent chain termina mark the end of the fragment and allow the sequence to be determined. What? What? Okay, determine sequence of nucleotides. That should probably be what it can be used for. Comparison. Okay, wait, actually, wait here. What can it be used for? Separating. Comparison can be used to determine relationship between sequences. Alright, cool. Sorry, I'm just getting a bit sidetracked. What can be used for determining sequence of nucleotide. Alright. Let's read this again. I don't quite understand, but hopefully the 3-minute video will make it clear. So in Sanger sequencing, the target DNA is copied many times, right? Copied. So you would use it. You would use what's it called? Gel. What? PCR to multiply it, I guess, right? That's the point, right? And then making many fragments of different lengths. Fluorescent chain terminator nucleotides mark the end of the fragments and allow them. What are chain terminals? What's this? What are those? Where did these come from? Alright, I'm just gonna write that down. In Sanger sequencing. In Sanger sequencing, the target DNA is copied many times, making fragments of different lengths. This sounds like the previous method. Different lengths. Hopefully I'll know it once. Fluorescent chain terminators? Is that what I said? Chain terminators? Chain terminators, nucleotide, mark the end of the fragment and allow the sequence to be determined. I don't get that.
I genuinely don't get that. It's lagging. That's always what I need, right? Please tell me it's not poor. I mean, please tell me it's no data. It's good. It's good. Cool, cool. It's good. It's good. It's good. Look at the end. It lagged. Fragment. You heard me typing, but it didn't type. I use a MacBook, guys. I know. I'm poor. What can you do about it? To be determined. Alright, cool. Okay, I think next. Okay, this image should make sense by the end of our discussion. Alright. Primal extended chain termination. Capture. I don't get it. No. Okay, fine. I will look at that. Okay, ingredients. All right, here we go. Here's like how is it done. So what do we need for DNA sequencing? Ingredients. Let me just. I need to memorize this. That's the thing about human biology. It's all about memory, to be honest. All right. Should probably. Oof, okay, what was I gonna do? So I need to blow my nose. I'll be right back. Right, cool, I'm back, sorry. Okay, how much have we done for time? Alright, 1 hour and 15 minutes. Not bad. So it's pretty good. Okay, a DNA polymerase ingredient. A DNA, why is that underlined? Polymerase enzyme, which I assume is the one that separates, no, the one that copies it, yeah. A primer, which is a short piece of single-stranded to the template. This sounds like the first one. I'm not. Is it just me? It sounds like the first one we just did. Template DNA and X as the starter. Yeah. Hey, this is like the first one. Okay, I feel like obviously it's different. Why would they? Starter for the polymerase. Starter for the polymerase. So, okay, we know what that is, obviously. We just learned about that. The 4 DNA nucleotides. I forgot what these mean. Oh, my teacher told me what these meant, but I forgot what they mean. Wait, DATP. If I just type this in. DATP. What is the meaning of this? Deoxyribonucleic triphosphate is a nucleotide using... Give me a straightforward answer. No, don't give me a straightforward answer. It's always got to give me a complicated answer. Why is that? Okay, 39%. Service recommended. That's always nice to hear. Okay, the 4 nucleotides. Okay, the 4 DNA nucleotides. I don't know what these are. I think they're the base pairs. Yeah, so there's A, there's T, there's C, and there's G. So DT, wait, is it capitalized? No, DT-ATP, DT-TTP, DT-GTP, what was that? CTP. I forgot what the DTP stand for. I'm so stupid. DD. Dio chain and the DDAP. DD. Oh, here we go. What is this? DNA sequencing. What are the DNA and DNA? DNA refers to deoxyribonucleic. Oh, okay. Wait, sorry. Now I remember. It's like, yeah. So this is convenient. So let's say this was A, right? This is referring to like the nucleotide with the phosphate and the sugar, like combined it with the base pair. So it's not just the base pair, it's combining it with the other bits, I believe. Refers to deoxyribonucleic nucleotide triphosphate. Yeah, phosphate. Yeah, phosphate. And what you got? This small green thing is a phosphate. Yep, called the temple DNA to be sequenced. Sorry, I cannot speak. The temple DNA to be sequenced. And that D-deoxy, what is that? D-deoxy, 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 is that even a word? Oh, there it is a word, alright. Or chain terminator, alright, so chain terminator, so we're going to learn what this is. Versions of all four nucleotides, that's very helpful. Versions of all four nucleotides. Can't type properly. Is the same. Each labeled with a different color dye. Alright, cool. Okay. Each labeled with a different color dye. Cool. Okay, DNA sequencing. The details, the method again, and that. And is it useful? Okay, so it's not that much. Okay, I thought it was a lot, but it's not that much. It's not that much. Thank goodness for that. What is this? Am I recording me? Alright, cool, it's recording me. That's great. Yeah, no, you saw my history. Oh no, my history has been nothing but study, which makes me look smart, but actually the only work I've done today is the work I've done on this stream. Oh well then. Alright, go. We've got the DNA sequencing. Okay, the details. As DNA is being copied, each nucleotide attaches by bonding to the hydroxyl group of the one preceding it. Okay. What? What the? Synthetic nucleotides without the hydroxyl groups are used to stop the elongation of... Okay, so the hydroxyl group is known as the third carbon. Alright, cool. So I explained before how this base has 5 carbons, and the third carbon is called the hydroxyl by bonding to the hydrogen. So it bonds to the hydrogen. Sorry, this makes no sense to me. Let's just go straight to the method, shall we? The method. I'm sure that was important, but my brain is too small to comprehend the complex words that is science. Okay, combine the DNA polymers, primers, DNA molecules, the templates to sequence, and the DNA. So just heat to separate, cool to allow heat to separate. This is basically the first one. Wait a minute, this is basically the first one. Okay, but it's okay. First one, but like different. Okay, maybe I should watch the video before going into this. First two were easy. This one's a bit complicated. Let's just write down the method, shall we? Sorry, hopefully my thing doesn't get. Okay, combine the DNA polymerase primer DNA nucleotides the DNA template sequencing to sequence. I hope my computer's making a noise like it's when it's overheating. Hopefully you don't hear that. Wait, actually I'm going to be silent for a sec and see if my audio actually picks it up. It doesn't pick it up, which is good. So it sounds loud, but it's kind of weird how it doesn't pick it up. Maybe it's designed not to the DNA template sequence and the deoxydeoxynucleotide. What is the deoxydeoxynucleotide? What was the deoxynucleotide? Oh yeah, this one. Synthetic nucleotide without the hydrocarbons are used to stop the elongation of the DNA sequencing. Okay. Okay, I know what that is now. So, where is it? Where is it? Where is it? Where is it? Here we go. Stops the elongation. Stops the elongation of the DNA. I think. Yeah. So each nucleotide attaches bonding to the hydrogen of the one preceding it. That's great. That's good to know. All right. The deal. Okay. The sequence. The nucleotides. Heat the specimen. Heat to separate the DNA. Cool to allow. So same as the first step. Well, pretty much most of it's the same. Allow the primer to attach. All right, pretty much the same. Heat to make the primer start synthesizing. Okay, this is where it's different. Heat to make the polymerase start. Oh no! What did I do? What was that? Make them start synthesizing. New DNA. So start creating new DNA. Okay, so heat to make the polymer start synthesizing. Okay, so heat it to make it move. At some point, that's very good to hear. Clearly they don't know what they're doing. I'm joking. Deoxynucleotide will be added to we are stopping the process. This process is repeated in multiple cycles. Okay, wait. Right then. So this is deoxynucleotides, which are also called synthetic nucleotides. So I should probably write that down. Wait a minute. Deoxy, right? Oh yeah. Yeah, so it's called synthetic nucleotides. So these are called all synthetic nucleotides. Versions of all synthetic nucleotides, which basically stops it. Okay, cool. That's good. Now what? Now the method again. Okay, by the time the cycle is complete, it's virtually guaranteed that a deoxynucleotide will have been incorporated at every single position of the target DNA in at least one reaction. That is, the tube will contain fragments of different lengths, ending each of the nucleotides in the original containing a dye compared to using electrons to determine the. Wait. What? What? I'm so confused. What is this? By the time the cycle is complete, it's virtually guaranteed. DNA sequencing. I still don't get it. By the time the cycling is complete, it's virtually guaranteed that a deoxynucleotide will have been incorporated at every single position of its target DNA in at least one reaction. That is, the tube will contain different ending at each and the final containing a dye. Oh, okay. I need to get my teacher to go through this. I really don't get this at all. I don't get it. I'm gonna have to ask my teacher. Oh, I'm gonna watch the video first. What's it used for? Determining sequencing, and it's also used for detecting hereditary disease. I don't know how it does that. Detecting disease. I'm just getting so tired. Is this around the time I get tired? Yeah, I started off strong, but now I'm getting very tired. Now I'm getting very tired. At least it's good. This is very cool, because usually it says poor, and that always annoys me. But now it's saying good, which is great. That's great. That's always great to hear. I'm not recording this, right? I'm just streaming. Okay, stop recording. Good. Okay, here's a disease, blah, blah, blah. Point mutation, which is basically another word for gene mutation, mutation, and small chromosomal mutation. Is that what it meant to say? Like deletion and insertion can be detected. Cool. Insertion. Partity, a minor process of genome between species. Partity and Martini? What? Paternity and maternity testing, which is basically a vital. I think that's all we do. I hope we don't need anything else. In genome comparison between species, evidence for evolution. Okay, I'm gonna watch the video, and this time I'm not gonna like skim through it. This time I'm actually gonna see whether it helps or not. Yeah, this is pretty much it. Okay, I'm gonna watch this because I don't get it. I might have to ask the teacher. But the thing is, we're being tested on this tomorrow. Why didn't I ask him today? Today we were given time to study this. My goodness, if only I had done this sooner. I could have asked about the Senga. What is that? Ow, my ears. Sorry if that blocks your ears. You may want to turn down the volume. I'm not going to turn the volume because I was done DNA sequencing. Boom. All right, cool. 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 So we're pretty much at the end of this. I just need to quickly get what this means. Oh no! I need to read, don't I? There's no voice. Why you do this to me? All right, here we go. Firstly, the DNA need to be sequenced. The DNA to be sequenced has to be amplified. So that basically means like it's copied. Heat is then used to denature DNA. I like this music. To produce a complementary strand and the temple strand for DNA. To produce a complementary strand and the temple DNA for DNA sequencing, right? Temple strand. The primer is then. Wait, what? That's way too fast. Primer is then annealed to the fifth end of the DNA, so over here. So that's where the primer is placed. Okay, so that's the primer. The primer DNA is then dispersed equally among the four. The primed DNA is then among the four vessels. Next, DNA pools is added to all four of the. Wait, didn't we just do that? Oh wait, no, that's the enzyme. Sorry. That's all the 4-deoxynucleotides, followed by all 4 DNA into each variant. Yeah. Okay. Oh, what? How am I meant to read that? Okay, specifically modified DNA are then added to the reaction vessel. All right, specifically modified. Specifically modified are then ordered. Oh wait, that's the same thing. Only one type of DNA is added to each vessel. Okay. That's the deal. Wait, but didn't we just add that? Didn't we just add that though? Followed by all fours of each. Oh, D. This just has a single D in it. This has DD in it. Oh, wait. I think I got something wrong. I think I got something wrong. Wait, I need to go back. There's one with the D and then the one with the DD. Where is it? Oh yeah, so this one only has D and this one has DD. Okay. I don't know. I still don't know the difference. Oh, so this is just DNA nucleotides. This is the deoxin. Oh, okay. So this wasn't the DNA. Okay, I need to quickly fix this then. D. Nope. This is just D. Regular D. See, videos are helpful. Videos are always very helpful. These are DD. Yep. Cool. So they first add the. Okay, they first add the DNA. The enzyme, and then they add the. I just realized I don't need the thing to be so loud. Followed by the what's it called? The DNA nucleotides. Oh, 4 of them. So like the A, the C, the T, and G. And then this is the deoxy now. So there's 4 types of deoxys, and you add one different type to each one. So this is what's happening inside. Okay. The polymers attach to the DNA, the DNA nucleotides to the temple strand with the primer, normally until a deoxy base is paired. Once the deoxy DNA nucleotide is base paired, the sequence is terminated. Okay, so yeah, again, so this is what like cancels the thing. DNA lacks a hydroxyl group at okay. As a result of the chain termination, DNA fragments of different lengths are formed across all the reaction vectors. What in the world? Polymer gel is used to sequence the DNA. Okay. Oh. Okay, I get it now. Okay, so what happens is, okay, so the 3 methods, they're not 3 different types of methods. They're all methods that help each other do the thing. So in order to do gel electrophoresis, right, you need to do 2 things. You need to do DNA sequencing, and then you need to do PCR, which replicate it, and then DNA sequence, which like puts it in a tube thing, as we've seen right there. And then you just do gel electrophoresis. Okay, it's all starting to gather up now. The pieces are all together now. Yeah, I get it now. So yeah, as we mentioned, these ones over here would be the smallest ones, and these would be the larger ones. Yep, we got that. Yep, we got that as well. What is with this music? Oh my gosh. Wait, what? Oh, I hope I don't need to know this. Polynographic, right? Okay, so the one with the P is used for. Oh no, is this something different? Was I wrong? Am I wrong? The sequence is read from the bottom plate. Wait, what? How did he read it? How did he read it? How did he read that? I'm so confused. This whole process results in the complementary sequence of the DNA sample. Okay, I don't get it, but I'm gonna have to ask. I will have to ask, but it's for determining ends with a complementary what? What was the video? Ends with a complementary sequence. Complementary sequence of the DNA sample. Alright, cool. 20. That's cool. Alright, cool. Oh, thank you for watching. Alright. Okay. We're done. I still don't get this. Wait, I still don't get this picture though. What is this picture? Okay. Okay, let me just quickly see if I can. I'm gonna skim this. I'm not gonna answer it. I just want to quickly skim through it. A minute sample of DNA has been obtained for paternity testing and to establish outlining 2 techniques in mind that can be carried out. All right, the one we just did, the second method, DNA sequencing, and before proceeding the DNA and PCR because we have to rapid development has been sequencing of a person. Lists some ethical issues involved in the DNA. Define gene evidence. Oh my god, for each of the techniques. For each of the techniques. Yep. Application, discussion, ethical issue. Okay, cool. Here we go with the original shot from Scott process. I don't know. Okay. Okay. What's gonna happen now is I'm going to log into my Language Perfect. I hope you don't see it. I'd rather not show it on the stream. Does it matter if you can see my username? I'd rather not. I'm gonna make it like that, so you can only see the RCC. Please tell me this is it. I don't remember me. Oh, it don't matter. You saw my username. All right, who cares? It doesn't matter. Hopefully that doesn't spell the end of me. Login to your school or organization. I'm not. Can you hear my computer? My computer's like overheating. Can you hear it? I don't know. The audio says it can't pick it up, but okay. So I've been given this task on Language Education Perfect, and my teacher says it's about 25 minutes to do. So by the time we're done, it'll be like, oh my gosh, we'll have time to do my literature homework. Okay, I'll do a little bit. I'll do like the planning of the literature homework, and then after that, I will do the rest at school because I'm gonna wing it. Oh, I was gonna say do it during lunch, but I have a meeting at lunchtime. How is this gonna work? This is not working. Why is this not working? Can I just reload? Yeah, reload. It's taking way too long. Wait, let me just quickly check my phone. Where's my phone? Here it is. What was I gonna check? Sorry, I just need to check something. What is this? It barely changed. Alright, cool. This is taking forever to load. Alright. Can I log in? Why is it still logging in? Oh, okay, here we go. We're in. Okay, so here's the thing. I need to do this. Alright, cool. Biotechnology review. Let's go. 26 minutes. Oh my gosh, that's a lot. Oh, thanks for the cheer, whoever you are. Amelia K. Alright, here we go. That's a lot. Oh my gosh. Well, here we go, guys. Wait, did I? No, I don't. No. Alright, yeah, okay. I'll just leave it like that. Okay, I'll just leave it like that. Okay, here we go. In this smart lesson, you will learn to describe biotechnology techniques which provide evidence for evolution, explain the purpose of PCR, bacteria, enzymes, and gel exotrophies. Outline how these techniques are used to facilitate DNA. Yes, we got that. 106 slides. All right, biotechnology and using living system for technology application. You will consider three biotechnological techniques: PCR, bacteria, and gel. Bacteria enzymes. What? What is this one? Is this? Have we covered this one? I'm not sure. Okay, PLC. PLC is a enzyme that used to make millions of copies of a DNA fragment. It is used to amplify the amount of DNA available to allow testing for DNA. DNA is a double strand molecule of A dances with T, C dances with G, hydrogen boss bases, sugar phosphate bases. Alright, cool. Yeah, I got this. PSA was first developed in 1983 by Nobel Prize winner American scientist Kary Mullis. He worked out that it was possible to artificially replicate the process of DNA replication to make copies of a DNA fragment. DNA works by a series of separating the DNA, then adding complementary nucleotides, which are joined to make two DNA strands, each with half of the original DNA. Semi-conservative. Alright. Cool. Yep. And lines. Yeah, we know this. PCR process. Mixture contains the piece of DNA copying primers, which are fragments of DNA which stick at the end of DNA when it opens. A heat-resistant enzyme. Yeah, we know that. Okay, so the enzyme that we have to use have to be heat-resistant. Nucleotides to form the new strands of DNA, and a buffer solution to maintain the pH. What? There's a buffer? What the? What, buffer? Wait, what? Okay, wait, whatever. Oh, just to maintain the pH. All right, I should probably add that. I should probably add that to my notes, actually. Where's my notes? No, that's not it. Where is it? Where is it? PC. Oh, man, this is gonna take forever, isn't it? Oh boy. Oh well, then let's get it over and done with, shall we? A buffer to maintain. Alright, cool. Oh, sorry, I forgot that it warns you not to have focus on this. Sorry, I need to quickly check what's happening. Anything happening? Nope. I've been going on for... What's my stream health looking like? Good, okay. Looks good for now. I'm not sure why this is still loading. Looks great. Okay, now I'm going back. Oh, now onto a 4. Alright, cool. Okay, superior separates the sun, denaturing, yes. Cooling to 65, yes. Next warm to 70. Okay, wait, sorry. So we cool it so the primers can stick. We denature it so we can separate the DNA. Next, the mixture is warmed to 70 degrees and a heat-stable DNA originally found in DNA, such as Thermus aquaticus, adds nucleotides to the strands. So that's the enzyme, known as elongation. Okay, sorry. So there's denature, there's annealing, and there's elongation. Is that the name of the processes? Oh, here they call it, okay, extension, elongation, same thing. So extension, that's when heats the content to allow the plant to synthesize new DNA. All right, cool. So this is when the DNA polymer synthesizes new DNA with the DNA nucleotide. Finally, the mixture is cooled to allow the strand to form two different DNAs. I didn't realize that. So it's cooled again, basically. Huh. The thermal cycles would then repeat over and with a single cycle only takes a minute. Each cycle is undivided. Alright, polymerase chain reaction. Yes, we got it. Denaturing at 64, annealing at 68, elongation at 72. Alright then. It has many uses wherever more copies of DNA are needed, basically. If we need more copies, we use PCR. They're given us to visualize and suddenly store by having more DNA in our sample to work with the ability to use investigate and increase immensely. For example, VSA is used to amplify samples of DNA to compare the DNA sequence of different organisms, which can be used to help and provide evidence for evolution. Alright, cool. That's cool. It's chain reaction. This one. Cool, I forgot it does that. Man, I hate that noise. Purely because I hear it to me. Recall who developed. Oh my gosh, it was Kerry Mullis. Kerry Mullis. Wait, sorry, can I go back to that? Kerry. I know that meme. Recall what DNA application means. Makes the science creates thousands of copies of DNA. Alright, cool. Recall what DNA is used for to amplify DNA. You just said that. Present the complementation basis from DNAs. I need always A with C with G and A with T. A-T, C-G. Alright, cool boy. Record the terms used to describe the steps of animation. Alright. Separation, that's denaturing. Cooling, that's annealing. That's annealing. That's elongation and application. Amplification. Cool. This is easy. Cool. Yeah, boy. We're actually going to do this in no time. How much have we done of this technique? How much of this have we done? Oh, 23%. So basically, okay. So it won't take that long. It's not going to take long. Sorry, I need to quickly go and do something. I'm going to go on a break. Sorry, I'm going to quickly go on a break. I'll be right back. Because I just need to get a snack. And I want to check on my little sister as well. Just wait. Sorry. I'll be right back. I'm sorry. I just need to take a break. Ugh. Sorry, I'm back. Sorry, if you just heard me chewing, I'm so sorry. I wasn't thinking just a moment ago. Okay, guys, this is looking good for us. I might get some sleep tonight. Alright, here we go. And the last one, the blue ones are usually the ones that takes the longest. How many questions? Just one? Alright. During PCR, there are cycles of heating and cooling. Using to understand of how enzymes are affected by temperature, discuss why enzymes are used in the process. Simple. Because thermophilic bacteria from hot backgrounds and hence are heat resistance, therefore want denature process, which would usually cease the function of regular enzymes. So I need to blow my nose out of it. Alright, cool. And I think that's it. The denaturing process, which would lose the carbon. Okay, I think that's it. Self-work. What is the current state? Okay. Enzymes function best at optimal. Yes, below the freezes, decreases. The activity decreases. Both the bonds begin to break. Depending on what enzymes so far, it can be for example. Live in hot environments and therefore adapted to higher temperatures and have a yep, pretty much. I'd say I did good. I didn't say optimal, I didn't say temperature. Oh well, who cares? Got it. Thank you. Let's go to the next one. Introduction bacteria. Okay, I haven't heard this one. I'm not sure why I haven't heard of this one. Well, how many is there? Okay. There's about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There's about 10. Right. Let's count down. Enzymes are proteins which act as biological catalysts, speeding up reactions. Enzymes are proteins which act as biological catalysts, speeding up reactions in cells. Right. When biochemicals, for example, provide evidence of evolution, we use enzymes to cut into fragments, stick bits together, and even make new DNA. The problem is, how is it possible to source large amounts of enzymes and have enzymes that function in a range of experimental conditions such as high temperature or extreme high? The answer lies in the world of bacteria. Alright, cool, that's interesting. Forgot that I had a mouse. I'm going to use the mouse instead of the chat pack. What time is it? It's exactly 11. And we've been going on for like 1 hour and 47 minutes. That's always a great thing to hear. I'm actually very proud of this. 33%. Let's go. Microorganisms are useful so they can produce rapidly in large volume by growing in larger tanks. Some bacteria grow in extreme events, such as thermophilic bacteria found in hot springs. On the other hand, these bacteria contain enzymes which are able to survive these conditions and do not denature at high temperatures or extreme of pH. This means that the enzymes can be used. Pretty much, yeah, we knew that. For example, PSR involves CT. If a PSR used to denature, news would be added every new cycle. New enzymes would have to be added every new cycle. Instead, PSR uses DNA presenting sources from the DNA, such as TAC DNA from Thermus aquaticus, or Pifrin DNA polymerase from polyphenyl's. These enzymes do not denature at high temperature. Okay, all I need to know is that they come from they're called thermophilic bacteria. DNA polymerase from the bacteria. And it's called TADEC, which is from Thermus aquaticus. I think that's all I need to know. Bacteria are also called to cut DNA into fragments. The type of DNA that cuts DNA is a restriction enzyme, right? These are like scissors for biotechnology. Cool. Restriction enzymes. Restriction enzymes were first developed when it was noticed that some bacteria seem to be able to fight off the attack of bacteriophage. On further study, it was found that bacteria possess enzymes which cut DNA into further sequences. Wait, so then what is regular DNA? What's regular enzymes? What do regular enzyme uses DNA? Okay, cool. Okay, yeah, good. Cool. Most restrictions are specific, cutting DNA to a specific enzyme. For example, the H for restriction enzyme recognizes the following sequence. It cleaves between, most restrictions are specific. It cleaves between the AAs on both sides. Oh, okay. ATT, AA, GG, ATT, GG, A. Okay, it cleaves on both sides. Yeah, I know this. Another group of bacteria that uses DNA ligases. The enzyme. Oh wait, what was the previous one called? Most are specific, cutting it specifically. Alright, these specific, but some DNA are ligases. These enzymes join fragments. Okay, another group of bacteria that uses biotechnology are DNA ligases. These are used to join fragments if they are matching, and just think of it as glue. So ligase is glue. So glue. So why wouldn't you want to join them together though? Here we go. Alright, recall why biology uses bacteria enzymes. They can produce in large amounts commercially. Yep. They survive heat stress, such as also found viruses. Yep. Okay. Recall what type of animal lives around volcanoes. Number 3. Recall what type of animal is used to form DNA strands in PCR. Okay, what type uses to form the new ones? So not the restriction that cuts it. DNA polymerase. That's the one. Recall what type of uses mostly. That's restriction. What type of use ligases? Alright, cool. Alright, DNA contains 2 long strands called sugar phosphate backbones. These are peach-colored strands. These are the peach-colored backbones. Alright, so that's the backbones. Cool. Resembling the rope of a ladder. Nitrogenous bases extend from each sugar phosphate backbone, making the largest step these. Alright, cool. The backbone of double helix contains sugar and phosphate, so phosphates are negatively charged. Alright. The phosphate groups are what give the DNA. Yep, we know that. We know that. Yes. Okay, so these are the sugar. Okay, so these are deoxyribose nucleotides, I think, and these are the. This is just a nucleotide. I'm so confused. DNA is contained with cell nucleus and protected by the nuclear membrane. The membrane stops scientists studying this DNA doing. To get around this, the DNA has to be extracted from the nucleus. The technique of DNA involves the use of chemicals to break down the nucleus and cell membrane, allowing the DNA to be released into the science where it can be isolated from the rest. And this allows us to study the DNA. Cool. Once the DNA has been extracted, amplified by a fragment using section, it can be visualized in an experiment known as gel extraction. Orders of each sample from largest to shortest. Since an organism's DNA is unique, the pattern created by DNA of each will differ. Okay, cool. Question time. Recall which part of the double helix is negatively charged, the phosphate groups. Recall what the job of a restriction enzyme is to cut. Recall where DNA is found in the nucleus. Cool, that's great. That was fun. Gelling is a technique used by scientists to separate DNA according to their size. It allows scientists to compare specific regions to DNA from different samples. It is used to forsake ants and companies ants to help are closely related in terms of evolution. Cool. That's cool. The first step of electrolysis involves making a gel. Sorry, not jail. Gel, I can't say that word. It's kind of like making jelly, right? Cool. Agarose powder is mixed with buffer solution and heated until it dissolves. The buffer solution resists changes in behavior when small quantities of an acid are added to it. Okay. Okay. Once it's cooled, edwin bromide is added to it. Make sure this chemical that binds the DNA so that it can be seen under UV light. What? This is, oh my gosh. Okay, once it's cooled, idiom bromide. Oh, I'm going to get these wrong. The agarose solution is poured into the special chamber to set. Combs are sent into place to create the holes or wells with the sample. Alright, okay, so they use combs to do it. Cool. Wait, go back to the second one. This one is added because they can be seen under ultraviolet. Use agarose because it doesn't change pH when acid is added. Each DNA is then piped into an individual process known as loading. Once a buffer solution is poured, either side of gel or combs are removed. In addition to each sample, a sample of known size fragments known as loading ladders is also, we talked about this in the video. This allows fragments of known size to be compared to samples being tested and their approximate size calculated. So compare and calculate. Cool. After samples are loaded, the chamber is wired up to electrical power source. Since DNA is negatively charged, the electric current is able to pull the fragments through the gel towards the positively charged terminals. Let me read that again. After samples are loaded, the chamber is wired up to an electric power source. Okay, so this power source is the one that starts the current. Since DNA is negatively charged, it will be pulled towards the gel towards the positively charged terminal. So negative to positive. The small fragments of DNA move faster than larger and therefore they travel further. Yeah, cool. In a given time period. Okay, cool. Recall what's the purpose of gel is to amplify, to merge, to separate DNA according to size. Recall the ingredients to create. Okay, here we go. Oh, wait. Oh, to create the gel? The gel? Okay, wait. Buffer. This guy. Agarose powder. That. No, done it. Okay, okay, so not the powder. We need the solution. And not the gel, just the powder. Oh, that's confusing. That's so confusing. Alright, to record the present ideas, facts, and electrode gel is created by mixing and heating agarose powder and buffer solution. Once the mixture can be added. Alright, cool. Recall why ethylene bromide is added to there. It binds to an influence on the thing. Yep, there we go. Okay, it needs buffer. It does not. It needs agarose powder. Oh, I'm so confused. I know it's number 4, definitely. It's either buffer solution. Yeah, there we go. Buffer solution and agar gel. Alright, cool. Recall what the purpose of placing comb to stop the sample node to create wells. There we go. For sample loading. Recall what loading means. Here we go. Pipetting the samples into the well. Cool. Explain why the power source is required in gel electrolysis. What? Okay, sorry. Explain why a power source is required in gel. To move negatively charged with the gel, to keep DNA, to move positively charged. This first one. Recall what size of DNA moves faster. Small fragments move faster. Alright, cool. We only have 1, 2, 3, 4, 5. Only 5 more, which is good. Once the gel has been run, the DNA fragments will be moved enough to visualize, although too small to view with the naked eye. That is where the actinium bromide comes in handy. Actinium bromide binds to even the smallest amount of DNA. It also fluoresces under ultraviolet. That means class. Okay, cool. When gel is placed under UV, the DNA can be seen. These fragments are known as bands. The bands of sample line up vertically with the well as the sample. The bands of each sample line up vertically with the well that the sample was initially loaded into. Yep, I got that. The bands represent the size of each DNA. Each fragment is made of a different number of base pairs. Small bands have less base pairs. Here we go. Small fragments have less base pairs and are able to travel further. This means that when the gel is filled, the larger size will be closer to the loading well, and the smaller one will be further away from the loading well. Cool. That's pretty much good. Okay. Allows DNA fragments to move across get gel at different speeds. So when the experiment is complete, then it can be viewed in order to determine the bromide can be considered. This makes it partially visualize the size. Cool. DNA fragments are known as what? Known as bands. Sorry, the fragments of DNA in the lens are known as bands. These bands, it's right there. The answer's right there. The bands in the size and lines up vertically with the well, with the sample that was loaded into. Wait, what? These bands vary in size and line up vertically with the well that the sample was loaded into. Yeah. Small fragments are able to travel faster through the gel, whereas some travel slower. This means that the band corresponding with the small ones are furthest away. Wait, what? Small ones, yeah. And corresponding are closer to the other. Easy peasy. This is easy. This is not taking 25 minutes at all. You are leading the forensic scientists on a recent assault case. You combined the crime scene for evidence and found traces of blood on the wall. You ran the evidence to gel accompanied by samples taken from a list of servers. Look at the image below. Which suspect do you think committed the assault? Okay, here we go. Okay, so suspect A and B. Suspect A, cool. That's suspect A, suspect A, suspect A, suspect A. Oh my gosh, suspect A. Suspect A, because all of the bases. Wait, what's it called? What are they called? All the bands match exactly with the evidence. Yeah. I'm writing more because they need me to write at least one paragraph. Different matches. So I'm just writing more because they need you to write more. I just said that twice. Okay, what do I think? Based on the evidence, I think suspect A may confirm this out. The band pattern of suspect A matches the band pattern on the sample. The band patterns of sample do not match the evidence. This indicates that suspect A had been at the crime scene at one point. However, it does not confirm they actually committed the crime. Oh, okay. So I'd say pretty good. Got it. Okay, cool. I think we just need the summary and this. Okay, this is for experts. If I don't know how to answer this, I'll just simply skip it. But biotechnically, such as DNA can be used to help identify closely related the more similar different species are biochemically, the more likely they have the common ancestor. Research and discuss if these provide enough evidence to conclude how closely I don't know. I don't know. I'm gonna skip that. I don't. Well, okay, wait. So this is how. Yeah. Wait. Let me think. Okay, so from what I studied last time, DNA is passed down to one animal organism to the next. Hence, if two species have the same DNA, have similar DNA, then they must have had a common ancestor who passed down that DNA to that organism. Using that, they can create a tree diagram, a family tree that can tell the story of evolution and trace back time. That's all I'm gonna say. Biotechnology using look at something. Oh, I forgot to mention biotechnology. As these evolving is likely to indicate how closely related. It does, however, make several assumptions, such as the rate of mutation being constant over time, whilst that is not always the case, as seen as rapid and to support the relationships suggested by there are several other types of fossil evidence, such as. Okay, yeah, I completely missed that. Hold on, I did not know that. I'll say three. Got it. Cool. Thank you. All right, here we go. Summary. Describe biotech. Yes, I got it. Yep, I got what both of those are. Used to amplify DNA using a thermal cycle, bacteria enzymes used in many techniques such as tech DNA polymerase and PLC. Restriction enzymes to cut DNA into fragments for electrolysis and ligands to join. Oh, okay, that's very interesting. So bacteria enzymes are used to cut, copy, and glue together, which is really cool. Electrolysis is used to separate DNA as a base. All these techniques are used in forming and helping study. All right, cool. We're done. We are done. Cool. How confident? I was pretty confident. How helpful did you? It was really helpful. Good summary of what we just did. Okay, that's good. I think we're pretty much done. Pretty much. Please tell me this is still going. Still going? Good. Okay, good. What we're gonna do now is we're gonna move on to literature homework, a bit of literature. Okay, so what I realized is tomorrow I'm going to school early, which means I do have time to work on this. So I will what I'm going to do is I'm going to just quickly do a bit of it and then finish it off tomorrow at school. So what is my literature homework? So basically I have to get a question, a literature, like an exam practice question, and do it on the Crucible. So what I'm going to do is I'm going to search up, I had practice questions, but I've left them away. I've lost them, sorry. Year 12 practice exam questions for literature. So I'm just going to search up a question. Yeah. Standard English P processing question. Here we go. This seems pretty reliable. Lots of smarts. Alright, cool. Wait. Close study of literature, 20 practice class. Here we go. 20 practice from Year 12 Standard English module. Close study of literature. Here we go. So I'll just use this. I'll put that as the thing. You can go check out this website if you want. Yeah, there we go. Okay, now which one am I going to do on the Crucible? Let's go. How has the composer of your text used conflict to express their key ideas? Oh, I know this. Oh, that one's so good. That one's actually so good. I might actually do that one. In your text, the relationship using memorable ideas. How is distinct qualities of your text describe your of its key thing? How is the composer of your text created an immersive moment? Are you serious? Oh, okay, fine, whatever. Only through the distinctive qualities do texts become memorable. Respond to this question. Only through the distinctive qualities. Yeah, I'm going to do the first one. Yeah, this first one's so good. Now, if you don't know what the Crucible is, wait, sorry, I need to go drink. All right, cool. So the express the key ideas. Okay, how is the composer of your text? So basically, if you don't know what happens in the Crucible, basically it's the town of Salem. It's about the town of Salem. If you don't know about the town of Salem, it's basically what happened was there was this one girl who wanted to wanted to. Sorry, I'm so tired. Who wanted to. Sorry. Who wanted to. Sorry, I just had a complete mind blank. Okay, so basically, there was this town who suspected that the town was full of witches that were like possessed by the devil. Basically, they went on this big witch hunt on hanging innocent people, killing innocent people, and none of them were witches. And instead, it was actually just a mischievous plan by some people to do so they could steal for greedy things, right? So basically, so to put it in a nutshell, there were people who were greedy of other things. They wanted this person's land, so what they would do is they would accuse that person of being a witch, all that stuff. There was this one person in particular. Her name was Abigail, and she was in love with this man, right? She's a in the book she's a teenager, but in real life I believe she's a lot younger. Basically, she was in love with this man, and then the man had a wife already. Though the man was already married, so what the Abigail did was make it so that the man's wife was accused of being witch hunt, and she had to go to be executed so that she could be together with the man. Now, obviously, she was a teenager, so she clearly didn't understand that wouldn't work, and that's clearly teenager work. Anyways, that's pretty much summary. If you haven't read The Crucible, you should probably go check it out. I will go right now. So what I'm going to do now is I'm going to just do the planning. Do the planning. Yeah, the planning should be good. Okay, I had notes on this. For quotes, right? Quotes. I need to actually write this. I don't know if I actually write this. Okay, sorry, it's so laggy. This is lagging again. Do I need any of this? No. I don't need this. I don't need. Have I done everything? Okay, there are practice questions to do on this website, but I feel like I've done enough for tonight. So I will take those out. How long have I been going for? 2 hours and 12 minutes. Okay, 2 hours. That's pretty good, actually. Pretty good. That's actually pretty good. It's actually pretty nice. Alright, let's go. Oh, wow, what is this? Okay. How is your thing planning? Okay. So there's planning and there's quotes. I think I was actually meant to write a practice essay for my teacher. My teacher wanted us to practice writing, and I don't think I can be able to do that by the time tonight. And by the time tomorrow, I'll be able to write like a rough idea. Hopefully she won't read out, like, we don't have to read out exactly what we wrote. Okay, but planning, let's start with the intro. Conflicts. Conflicts. Every story has some sort of conflict. Most can be used, and most are used to express key ideas of the author. An example of this would be Arthur Miller's book novel. Was it prose? His novel, The Crucible. No, wait, play. Sorry, this is actually a play. It's play. The Crucible. All right, Crucible. This play depicts the story of the town of Salem, who, in moments of hysteria, accused and hanged innocent people will be witches. The main conflict of this story is between the accusers and the accused. Okay, so what are the key ideas that this play expresses? Well, what I read into this is that it was very anti-religious. Anti-religious. Right? Right, think about this. Think about it. Hear me out here. Why is it gray? Here we go. Planning. Right? Because the main conflict that occurs in this story is a conflict that can only occur in a religious setting. So these are people who believe that the devil actually exists, people who believe that God exists, and hence that is why they believe that witches existed. Because if you weren't a Christian, you probably wouldn't believe that witches existed. Hence, if anyone was accused of being a witch, they would be considered stupid. In the Bible, it does actually refer to witches. I forgot which verse it is. I forgot what it says about them too. But basically, witches, people, only non-believers wouldn't believe in witches, hence this hysteria would have never hit them. That's why it's anti-religious. Another idea that it expresses is, what is it about? Between conflict between authority and those below them. And the key idea being that, what was it? Everyone's human, maybe? Everyone is human, no matter the authority, I guess. People can make mistakes. I think. Because loosely, I think, yeah. People can make mistakes. I think because loosely, you would expect the authority to be everything right? Because you expect them to be right, because they're leading, right? You want them to always be right, but they still make mistakes. And the mistakes that the authority makes is that they hang innocent people. Hence, but here's the thing: they don't want to admit. They don't want to admit that they were wrong after hanging several people, and hence creates conflict. This shows the main idea that, well, because the authority were like very hysteric, very I don't know, man. There was conflict between Proctor and the people who used the Bible in a twisted manner. And then there was, I need to find quotes later on, but, and then what else? So there's the anti-religious, there's the fact that people can make mistakes, like even people of high authority, they can be wrong, sometimes very condescending. What's another conflict? Ooh, conflict between Proctor and himself. All right, hear me out here. Okay, Proctor. I don't know if this counts as a conflict though. I mean, there's conflict between Proctor and his wife. Proctor. Yeah, so himself, his wife, and Abigail. Proctor is a changed man by the end of the book. Before he dies, he trying to keep his honor. He hid the fact about cheating with Abigail, even though it would have stopped Abigail's plans. So he's trying to conflict himself, right? He's cheating on this, like he has a conflict with himself, and then he wins his conflict, and he realizes he he is confused on what it means to 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 I have nothing but, he talked about his name near the end, like I have my name and nothing else but my name. To have an honorable name, what it means to have, it means thing to something, It means to, like, to be honorable, to be considered heroic or have a heroic death. Like, why was his death so heroic? Because he stood up for what he thought was right. And finally figures it out by the end of the play. And there's conflict between his wife, Proctor, and his wife, Proctor versus his wife. What key idea? Proctor versus Abigail. Foolish teenage knowledge, foolish knowledge, or the greedy, unlogical reasoning of teenagers. Abigail thought that if Proctor's wife died, Proctor would go to Abigail and marry her. And then his wife, the wife knew that Proctor cheated on. Her, but kept quiet, even lying. To defend. So I just defend. Whenever I'm writing like this, I always go silent. I'm gonna try. Okay, so he tried to. So if you guys don't know, it's a plot twist. You should probably go read the Crucible yourself if you're interested. But in order to defend his wife, in order to defend his husband, he defend Proctor. She lied. Defend Proctor, which ironically, however, made the situation worse because Proctor had already confessed at that point. Had already confessed before at that point. Okay. So Proctor and his wife. This probably goes. I don't know what the conflict is. They both kept quiet about it. They both kept quiet about the adultery because first, back then, the punishment and honor that would be lost would be severe, and so they continued as if nothing happened. Okay, I don't know, man. I don't know. Okay, I think I'm gonna stop here. I'm gonna sleep. I need to sleep. What time is it? 9. It's 11:33. So I should probably go get to sleep. I'm gonna. I think there's nothing really left to say. Tomorrow, I hopefully want to write a short story, so that would be more interesting. I'm still gonna call it a boring stream, but hopefully it'll be somewhat. Actually, I won't. I'm just gonna say writing short story. I think I'm just gonna end it here now. Well, 2.5 hours. That's pretty good. Pretty good. I think that's all. I will have to quickly summarize through this. Do I have to? No, I'll do it in my free time. I was gonna quickly go through this, but you know what? I'll leave that there. I have that website, and I think that's it. So yeah, guys, if you've watched up to this point, which is probably nobody, I'm probably still talking to myself, which is good. I'd rather not people watch this boring stream, but anyways, I'm gonna leave it here. Should I wait 30 seconds just so it's exactly half an hour 2.5 hours. That's pretty. Is that my longest one? I think that's my longest one. But I don't know. So I think I'm gonna again. I kind of want to stretch it to 30 minutes. So get that free ad revenue. I'm kidding. I'm kidding. Okay. Anyways, yeah, I'm gonna go sleep now because I have to test them all. Wish me good luck. The previous test was pretty good. I still haven't gotten it back, and yeah, I've got like 5 seconds. But I got to answer every question, which is good. 5 more seconds. I'm gonna finish in 3, 2, 1. See ya. Ah, come on. No, 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 no. There we go.